Morning, everybody. Welcome to our grand rounds. Today is October 1st, 2020, and we have the privilege this morning of having Dr. Pouratian from UCLA talk to us about the early results of a visual cortical prosthesis for patients who are blind. Just by way of introduction, Dr. Pouratian is a professor of neurosurgery at UCLA. He did his medical school actually at UCLA, where he earned an MD and a PhD, and was actually quite prolific in both his scientific work and his clinical work. Currently at UCLA, he's vice chair of Academic Affairs, chief of the Division of Functional Neurosurgery, and his practice interests include neuromodulation, radiosurgery, peripheral neurosurgery, as well as neuro-oncology. As a neurosurgeon, neuroscientist, and he has a unique skill set of really to try to, I think, fuse what's going on from a bioengineering perspective. And I think, I've known him for years, and he's probably the smartest person that I know, if not have ever met. He's got several NIH grants, and everybody knows who he is in functional neurosurgery, and I'm really proud of the work that you've done. Nader, thanks so much for joining us.
Thank you, Rod, for the invitation, and thank you for the opportunity to speak with all of you again. I'm just sorry I couldn't be there physically to see everyone and spend some time with all of you. My last visit was great, and hopefully we'll get to a time where we can actually see each other in person again sooner than later. But for now, we'll Zoom, and we will go talk about some of my work. So thank you again so much. Let me just pull up my slide here. As you heard in the introduction, I'm going to be speaking about some of the work we've been doing over the last several years on developing a visual cortical prosthesis for the blind. This is work that came about through a encounter of convenience in that I had one of my colleagues from graduate school who was working for a company called Second Sight. She was the VP of research and was looking to start developing a cortical prosthesis. And she reached out to me at UCLA, and it was a topic that I was interested in as well. And we started a collaboration 6 or 7 years ago that progressed rapidly to a first-in-human trial. So I'm going to show some of the work that we did to build up to that and what we've been doing with these people who've enrolled in our trial and we've been working with closely. So I want to start with just acknowledging that the field of neuromodulation and brain stimulation is alive and well, and sort of the mainstay of therapy within neurosurgery with brain stimulation is deep brain stimulation for movement disorders such as Parkinson's disease and essential tremor. We've seen an emergence of brain stimulation on the non-invasive side, particularly for psychiatric disorders with transcranial magnetic stimulation. But on the invasive side, we've had lots of trials looking at lots of different applications. And unfortunately, we haven't had too many new indications, apart from some relatively recently, including epilepsy, which is our newest indication. But a lot of work has to be done, and it takes a lot of effort. And I think there's a lot of promise to develop these therapies, and it takes work and collaboration across multiple disciplines. So one of the areas that I'm focusing on is blindness. And the estimating the amount of blindness, the number of people with blindness in the world is actually quite difficult. And I've learned a lot about it throughout this process. We often talk about blindness and we assume that someone who is blind just can't see anything. And it turns out most people who are blind see some shadows of light or can see some general movement, but they have no definition. But there's probably about 39 people worldwide who are legally blind, 8 million who are blind due to unpreventable causes. Retinitis pigmentosa is highlighted here as less than 500,000 people with that. And the reason I highlight that is because it's the only indication or diagnosis of blindness with an indication for an implant to provide artificial vision. And that is actually an implant that was created by Seconds. I will take a look at that in just a moment. But then you move on to something like macular degeneration, and there's 2 million people, and other causes, there's 6 million people. So there's a lot of unmet need in this group in terms of providing some kind of visual restoration. Now, as I said, retinitis pigmentosa is the only indication that there's a device. This is the device. It's made by the company that I collaborate with. It's an array that goes on the back of the retina that is then coupled to an electronics can, which wirelessly connects to a set of glasses, a video processing unit, and there's a camera on these glasses that translate the signals and stimulate the retina directly. I did not work on that project, but that was the background of the work that moved us forward to the brain prosthetic. But the problem is for this to work, you need an intact retina. You need a retina that can be stimulated. There's a lot of people who have blindness from other causes that don't have an intact retina. And so the unmet need was still huge when we started this project. And we thought, maybe going directly to the brain could provide a solution. But there's a lot of challenges there. Of course, our overall goal was to develop a cortical prosthetic for people who are previously sighted, because we want their visual cortex to have been developed. But these are people who have no or bare light perception, meaning they're not the ones who see shadows, but they see blackness or they see lack of any light. That's a hefty goal. And in terms of what kind of vision we want to provide for them, it's something that we've been evolving our understanding of, and we'll speak about that in just a moment. But there's other major issues. We want to make sure that we have a safe implant. Our biggest concern is if we're stimulating a large area of visual cortex that we could cause seizures. We wanted a practical solution, something that I was very committed to when developing this approach, to have something that could be adoptable, meaning not something that only 3 surgeons in the world can do, but something that if it works, that we want many providers to be able to provide this and for it to be useful for as many patients as possible. And then we wanted it to be clinically useful. And this is a very difficult area to define. And I think we're understanding more and more as we go through the process. But people ask me, when are we going to make people see like we do? And I don't know the answer to that. I don't know if we will ever accomplish that. But we have to understand where these people are coming from. They are coming from no light perception whatsoever. So they don't know anything about their visual environment. So if we can give them some kind of perception to know where a doorway is, to know when people are walking in front of them, to know where edges are, an edge of a table, a hallway, that can be incredibly useful when you start from absolutely nothing. And the other major challenge that we saw was that with brain stimulator, the highest channel count when we started this was only 8 channels. And we realized that if we're going to stimulate the brain with any kind of sufficient amount of resolution, that we're going to need more than 8 channels. I should say that this concept of brain stimulation, although as we were developing it and thinking, how do we adapt prior technologies to meet this new need, it's not that this concept of visual cortex stimulation is completely new. This is some work from Brindley and Lewin, and Dobell also did some work in this space. This is from a couple decades ago. but showing that, you can put an array of electrodes. This is frontal, that's occipital. So we're looking at the medial occipital lobe. The black line is calcarine sulcus. And they basically show that, well, you can stimulate the visual cortex in these different areas, and someone can report that they see a phosphene in a certain area, and it maps about closely to what you'd expect from what we know about visual cortex organization. So this has been done, but when this was done, these were done with externalized electrodes. So electrodes with a pedestal either on the side of the head or with wires coming out of the head, which is not a long-term solution. And so the durability and the ability to do this for the masses was not completely clear. I also want to emphasize that it's not just from a couple decades ago. There's still active work in this area. This is work from Stanford showing these are in epilepsy patients who have electrocorticographic arrays over the visual cortex. And they did some elegant experiments I'm not going to get into, but basically showing that you can predict where patients will see a flash of light, both based on their visual cortex map that you can get from atlases, as well as looking at how the brain reacts to light when it's showed to them. So they did some very active research. But again, this is in sighted subjects. And so people who can see may not perceive things the same way as a blind subject. So there were a lot of questions that were unknowns. And as we got into this, the first thing we wanted to do was, just prove to ourselves before we take on this huge endeavor, can we chronically implant a patient with an array, stimulate the visual cortex, develop Phosphenes or have them see Phosphenes and be able to demonstrate some reproducibility. And so we did this. We had a patient, we decided to take an off-the-shelf device, which is the NeuroPace RNS device, which is an epilepsy device. And we wanted to implant it in a patient who was blind. This was a 30-year-old woman with a history of bare light perception due to this relatively rare diagnosis of Vogt-Koyanagi-Harada syndrome. But the goal here was, can we implant a device over the visual cortex, have her see things consistently, try and understand the dynamics of stimulation, what it would take to actually have people see things. So we did this surgery. We put two 4-contact arrays over the visual cortex. So here's the calcarine sulcus and the two 4-contact arrays. We have 3 above the calcarine sulcus, 5 below. And there's a couple things I want to highlight here. What you see in each of these small graphs here is what the patient could indicate to us in terms of what she saw when we stimulated the brain. And there's sort of a large area that she would swipe over and show us by touching a monitor. And what you see is that it's pretty consistent. If you stimulate here, you get a flash in the bottom left visual field. If you stimulate at this area, it's more medial. Below the calcarine sulcus, you get visual perceptions above the horizontal meridian. And what you'll notice is that these are month 8 and month 19. So over a year and a half, we get extremely consistent visual perceptions with stimulation. The other thing you notice is that stimulation at different areas produces different perceptions. So it's not that every single stimulation produces a dot. Sometimes you get a large oval, sometimes you get a smaller dot. It changes. So there's different shapes and sizes depending on when you stimulate, but there's consistent localization. Now we got into a bit more of the details because what we wanted to do was, as we were thinking about how to design the cortical prosthesis, we wanted to know what kind of charge characteristics the device needed to have. How much do we need to be able to stimulate? What frequencies do we need to stimulate at? What are the design characteristics that we needed? And so we went through a series of experiments where we stimulated at different frequencies, different amplitudes, different pulse widths, many different parameters. And we're able to start plotting and saying, okay, if we stimulate at 10 hertz, in order to get this, these are thresholds for visual perception. in order for the patient to be able to consistently say that she saw something, we had to stimulate at 5 milliamps. But if we went up to 20 hertz, we only had to be at 3.5 milliamps. And so you can imagine how we produce these. These are two different electrodes. One is A1, one is A3, but very different, very similar patterns where you have very high amplitudes at low frequencies, and then lower amplitudes at higher frequencies. And this is true with frequency or with pulse width. We see similar things if we look at the charge per trial, meaning the amount of charge that we needed per trial was relatively constant to produce these curves. The important thing about this is basically we're working on a macro scale. We have electrodes working at the 4 millimeter size. But this exactly recapitulates the patterns that we see if we try and directly stimulate a neuron and get it to fire an action potential. So longer charge durations, there's less current that's required. So we need less current the longer the charge duration. And also there's somewhat of a plateau. At some point, no matter how long your pulse width is or how high your frequency is, there's some minimum current that you have to be able to inject into the brain. Why is this important? It gave us a lot of ideas about how we need to define our stimulation parameters as we thought about what to go to next. So this was a big undertaking. It was a little bit interesting. I'd never done anything like this to really implant someone purely for the sake of science, purely for the sake of device development. This was never going to be a therapeutic device for this patient, but she was eager to participate and eager to help us advance this field. And it was really critical to get us to FDA approval for our clinical trial. So we're indebted to her for her volunteerism and her commitment and passion to helping the field. So when we finished the study, or when we, at that time, I took this from one of the slide decks that I had from early on talking about all this. Those are the conclusions that we had. But we had a lot of questions. We said there are further studies about phosphene modulation, like how can we make the shapes of these phosphines different, the size, color was an interesting thing, because these are usually black and white perceptions. Temporal factors, sequential or simultaneous stimulation, and frame of reference and head and eye position. So the interesting thing is, and you'll see as I get to towards the end of the talk, these are all issues that we've actually started to tackle because they're important. They're important for actually making these clinically meaningful devices for patients. So the device we have worked in was developed by the company that I collaborate with and for which we have an NIH grant to conduct these trials. It is leveraging this technology, which is the Argus 2, which stimulates the back of the retina, which I showed you before, but taking the same electronics, but now adapting this electrode so we can implant it in the brain. And we went through multiple design iterations. We did cadaveric studies. We did animal studies to demonstrate safety before gaining the FDA approval to go ahead with our first in human trials. I won't go into too much detail here, but I did just want you to be able to see what the device looks like sitting on the fovea. This is one case where I can say that I think the neurosurgical procedure, the brain surgery, is actually easier than the ophthalmological surgery. This surgery is actually extremely difficult to get this electrode array to sit on the back of the retina. And so we developed it differently for the brain. So this was our adaptation for the brain. This is what we call the Orion I implant. Same idea, instead of having the electrode array sit on the back of the retina, it's designed in a triangular shape to sit on the medial aspect of the occipital pole. It's connected to a can with its electronics that delivers the stimulation. It's externally powered and controlled through an RF coil, so all of this sits under the skin. It's implanted. The patient has glasses that have a video camera that then couple through the skin with this device through the RF coil. And there's a video processing unit. This takes the signals from the camera into the video processing unit, translates them into stimulation patterns that are relevant for the individual patient, and then communicates with the RF coil to deliver the stimulation. So we're taking pictures of the outside world, translating them into a stimulation pattern, then delivering them to this device that directly stimulates the visual cortex. For the first in-human trial, we only implanted one side of the brain to mitigate any risks associated with it. This is one of the images from our study. We have implanted 6 subjects, 4 of them here at UCLA, 2 of them at Baylor with my colleague Dan Yosher, who's the principal investigator at Baylor, and his outstanding team that I'm going to show some of their work. But it's basically a parasagittal craniotomy. Insert this electrode over the posterior medial occipital lobe, over the calcarine sulcus, which you can see here. Here's the calcarine sulcus. You can see the array would fit in right here, and then a skull-mounted device. These are the subjects with the duration as of May 15th, 2020. We had a bit of a pause because of COVID, like a lot of people did. You can see an array of patients, mostly male, one woman, and an array of causes of vision loss. And that's important because it's not specific to a specific type of blindness. But we've had people implanted anywhere from 16, now 20 months, up to 30 months or more. So first things first, we have 60 electrodes on this array. So remember I said when we started, the highest channel count was 8. Now there are electrodes with, or systems with 16 channels for deep brain stimulation. We're up to 60 with this device. And we wanted to see, with the approach that we were using, how many of those 60 contacts actually produce something that the patient can see. And it's actually an extremely high number. So anywhere from 56 out of 60 up to 55 out of 59. So there's a very high number of perceptions, which is fantastic. That gave us a lot of options in terms of how to control the stimulation, how to deliver the stimulation. And this is something we've been watching very closely in terms of how persistent those are. And then we get into testing and how do we know if we're actually helping these patients. I want to, again, it's really important to put things in perspective. We all know the scale, the vision scale, where 20/20 is what you want to be. You can see things from 20 feet away that you should be able to see at 20 feet away. And 2200 is where we are legally blind. And now we are way below 2200. So someone can say they're blind and be 2200, but we're talking about people that can't count fingers, that can't see hand motion, that they have bare light perception or they have no light perception. So we're looking at the very bottom of this scale. And so the tests that we're doing are looking at direction of motion, square localization, and I'll show you some of those things now. So this is one of our subjects doing a square localization task. He is completely blind. He has no light perception without this device. And his job here is to tell us where the square is. This is, all of our participants have figured out strategies of how to do this. And so a lot of them use this strategy in particular, where they scan the screen. They're looking for where they see bright spots of light depending on their head position. But you can see he does an outstanding job of doing this. He's able to pick up on that square. He's able to point to it almost perfectly every single time. And really has no problems with this. So we're going to I just wanted you to see what this is like. And then go to the next slide. And here's an example of how the square localization would work. If the patient has the system off, you say, try and point to the white square. And here are all the answers relative to where you'd expect the square to be. And you can see the pointing is all over the place. Whereas with the system on, it converges onto that square. So definitely they are able to use the device for localization with a combination of the device as well as head motion. We see this is their average mean error with the device off, which is pretty high. And 5 out of our 6 subjects have significant improvements in their mean error with the device on. There's this one patient who has, there were some reflectivity issues and some training issues. Patients really need to learn how to use the device. And so we've been working with that patient to improve performance further, although it's clearly not a significant improvement there. This is a different task. This is a direction of motion task, and I don't have the video here, but this is, imagine sitting at a screen and seeing lines move in a certain direction, one way or another. And the job of the patient is to indicate which way the lines are moving. And you can imagine if they have the system off, they have no light perception, they are not going to know at all which way the lines are moving. And so there's a pretty even distribution across all the different responses. This is how many degrees off they are, versus here, this is with the system on. You can see that there's a convergence on basically 0, that the error approaches -20 to 20 in terms of localizing direction of motion. And again, here you can see system off versus system on, significant improvements in every subject in terms of improving their direction of motion perception. Now, all that is, that's good for testing. And really what's important is how do the patients describe the use of their system? Do they find it useful or not? And so we have rehab specialists, low vision rehab specialists who work with all of these patients and or their families who give us sort of snippets of life in terms of how they use the device. So I'll just read through these because I think they're telling about how useful these can be. So the first one, he was able to find the cue ball with no problems on the table. Excuse me. He can tell the cue ball from the blue 10. So this is, we're looking at a pool table. Here's one, he was able to see cars parked on the side of the street, opening in the sidewalk up into driveways. She was able to correctly determine whether I was traveling left to right. So these are people who can't see anything and now can tell when people are moving. Ordering patterns of small checkers, big checkers, and white cloth. So they find success with tasks for everyday tasks. I do want to show you one more video.
By moving his head, Jason can distinguish light areas and objects from dark.
Little white dots on a black background. It's like looking up at the stars at night.
Only the world's second person to receive the device, Jason is helping the technology to grow more useful as he learns what each flicker of light represents.
If someone moving across the room, walking past me or walking away from me, or the light against the wall, it was just amazing to have some form of functional vision again.
Now he can perform everyday tasks he couldn't before, like sort laundry and safely cross the street. And with a little practice,
I'm closing the camera to show you that I'm not cheating.
Jason continues to expand what he can do.
So that shows you a little bit about everyday life with the device. It doesn't look fancy yet, and we're improving, the device is being improved in terms of its externals, but clearly shows some improvement. Now, I wanted to get into a little bit of the challenges. So it's not all amazing. There's things that we learn after we do these types of studies, and eye position is actually extremely important. We think about the brain. We all know that the left side of the brain sees the right side of the world, and the right side of the brain sees the left side of the world. But what you might take for granted is that when the eyes move, the expected left and right side of the world is different. And what we saw with our very first patient, the one that we implanted with the off-the-shelf devices, when she saw a perception, she would look towards it. And the more she looked towards it, the more it chased away from her. So she could chase these perceptions away. And what happens is if the eyes move, the relative the brain's interpretation of where that stimulation is occurring also moves. And so we took on a series of studies with Avi Kaspi, who's from the Jerusalem College of Technology, looking at this with eye trackers and looking at how patients interpret where they see things relative to eye position. And there's some really interesting results here. So this is with stimulating a single spot in the brain. And you could see these are the responses of the patient in terms of where she indicated that she sees the light. And the arrows tell you which way we were telling her to look. So we said, you know, look to the left and we'll stimulate your brain. Look to the right and we'll stimulate your brain. Look up, we'll stimulate your brain, down, and in the center. And it distributes quite nicely according to eye position. And we can look at, you know, where the perception is relative to eye position. And in fact, if we backtrack and we say, well, what if we compensate for all that eye position and we now map the outside world to our visual cortex world, we can get a very consistent localization of where that percept actually is. So what we found is that eye tracking is probably extremely important for really interpreting where people see things, and that it's probably an important part of the technology. We did this with the Orion implants as well. So we see the same thing here, that if we stimulate two different electrodes and they're far apart, but we don't control for eye movement, you sort of get this blur of where patients might say they see dots. They're all in a different line. Same thing here. These are even closer in terms of the perceptions. But if you account for eye movement, you now get two very clear areas of separation in terms of where people see things. So you'd think it'd be easy that you could tell people to, well, we are so, as human beings, we're constantly scanning our environment. We're used to moving our eyes. Turns out blind people also do that. We respond to external cues. And so telling them to just look ahead and move their head is actually a pretty difficult thing to do. So this idea that we need to track eye movements to interpret what's going on and how they perceive things is actually extremely important, something we're working on to improve the system further. And that highlights that you know, we have some challenges. You know, we have what we would call a lookup table. When there's a video camera that gets a picture of the world, we have to figure out which electrodes to stimulate on the brain and how they map to the world. But it turns out this spatial mapping is actually quite complex. I just showed you one example, which is eye position. But also we have to account for the fact that these people are blind and we're relying on a motor response, and their hand-eye coordination isn't as good as a sighted person because they don't have the visual feedback to know where their hand is. There's other factors like how far the monitor is from the patient. The stimulation parameters can change the perception. And so this is another area that we're working on to better define where it is exactly that they see these so we can improve the patterns of stimulation that we're using. You know, we can get an experimental map, meaning, you know, ask them to tell us where they see dots. We can also look at the atlas and say, you know, where do we expect those dots to be? And they're not always in the same spot and they don't have the same distribution. So what we're trying to do is develop these optimized templates that take into account what we know about the brain, take into account what the patient tells us, but knowing that what the patient tells us has some limitations, and then testing which of these templates are the best. I want to end with one final story, which is work that was done primarily by my colleagues and partners at Baylor College of Medicine with Michael Beauchamp and Dan Yosher, Bill Bosking, and all those who are listed above with this publication that was in Cell. And this takes into account a couple other concepts, but again shows you how it's not as simple as just developing a surgical device, plopping in the brain and turning on some electrodes. And there's really two innovations here. One of them is this idea of dynamic current steering, and I'll show you what that means. But then also it's two. One is dynamic, so stimulating over time, and then current steering, which is stimulating electrodes at the same time. And I think this picture really highlights this the best. If you were to take your hand and someone just touched your hand in the shape of a Z, it'd be really hard to tell that it's a Z. But if you were able to draw out a Z, you'd probably know that it's a Z much more. And so it's the same thing, this idea that we need to draw things out in the brain, not just stimulate the brain all at once. So a couple of concepts here. This is 2 electrodes on the brain, just showing the dynamic current steering, meaning if you turn one of them on all the way, you'll get a certain perception. You turn another one on all the way, you'll get another perception. But if you turn them on at the same time at different levels, you can actually steer between those 2 points. So you can get these virtual areas of brain stimulation that can help improve the perception of light or increase the resolution of the stimulation. And what you can do then is imagine you have 5 electrodes in a row. You can turn one on as one is turning off. You can turn the other one on, and you get this dynamic stimulation. You get this dynamic current steering, and you can get someone to actually draw a line across the screen based on this dynamic stimulation. And a line is interesting, but what if we took it a step further? So again, this is our first patient with the off-the-shelf device, and it's not pretty because there's only 5 electrodes that are being used here, but you can see that she can draw out shapes that look like letters, an N or an R, upside down U, a V in this case, a W or a Z. And this is again with dynamic stimulation and then current steering. So this idea that you can't just turn turn them all on at the same time and hope that they see something. We actually have to deliver it over time. And finally, this is in a patient with the with the Orion device, again showing that we can actually get much more resolution. So you can draw out a W, an N, an M, and a U with a lot of consistency with the patient not knowing what they're supposed to be drawing out, really showing that you can get some complex forms with this dynamic current steering to further enhance these starry night pictures that you heard the patient describe. So we don't want to just have them see flashes, but start to develop these more complex phosphenes. And that's what we're working towards now. So I'll conclude with, when I started this project, I had this vision, no pun intended, of providing providing vision to people who are blind. And I don't talk about giving people vision now. I talk about giving them artificial vision. I am a strong believer that this device is not meant to replace vision, but is to give them a different kind of vision. And they need to learn, our brains are powerful organs, and people will learn how to use it. The device is safe. I didn't include, and unintentionally, a slide about side effects. There's one patient who's experienced seizures. We've been able to work around it and avoid further seizures, but we have had one patient who had a seizure, and other people who've had expected issues of headaches or pain or some discomfort. One patient reporting vertigo as well, although we think it's unrelated to the device. In any case, we really do feel we're very encouraged by these early results. We're still studying these patients, trying to understand the stimulation further, how to optimize it, and are working towards developing the next phase of trials to implant more patients and develop this therapy. I do want to just make one plug for some other work that we're doing, but I think it goes along the same line. The field of neuromodulation is challenged by this idea of putting electrodes in people's brains and hoping that it works. And I think this era of just putting it in, turning it on, and seeing what works, or if it works, is probably, we're past that. We're much more interested in defining how stimulation works. You just heard me talk a lot about how do we optimize stimulation. This is a project I'm doing with a good friend of mine, Samir Sheth, who also went to UCLA Medical School. He's an MD Ph.D. He's a neurosurgeon. He works at Baylor as well, coincidentally, has nothing to do with the Vision Project. But we're implanting patients with depression. And we're putting electrodes in the subgenital cingulate and the ventral capsule, ventral striatum. But we're also treating these patients like you would an epilepsy patient and putting stereo EEG electrodes in their brain so that we understand how to optimize the stimulation. We want to understand what the effect of stimulation is on the brain and which stimulation parameters will work best. I chose not to present this today, but it's a similar concept overall, that you can't just put electrodes in someone's brain and turn it on, and if the trial fails, well, that's too bad. We really need to understand the science of the brain, the science of brain stimulation better, so we can develop the new generation of therapies. We've got lots of these clinical trials from this type of reverse engineering. We have the brain stimulation for blindness, DBS for depression that I just talked about. We have another trial for depression. We have a DBS for chronic low back pain with my partner, Dr. Bari. We have a VNS for cardiac arrhythmia. There's a lot of work going on in neuromodulation, and there's really there's really no organ system in the body that isn't affected by the nervous system. So I think there's a wide open space for us to improve the human condition through neuromodulation. I want to acknowledge the people who work particularly on all these visual cortical prosthesis trials with Second Sight, Al Mann Foundation, which is the parent company that Second Sight spun off of, our team here at UCLA, Jerusalem College of Technology, and then Baylor College of Medicine that did that last study that I shared with you. And of course, everyone else in my laboratory who keeps me afloat in all my collaborations. And I'll stop there. Thank you very much. Happy to take questions.
Hey, Nader, that was terrific. So how many patients total did you have? That one slide, what is it, 6?
Yeah, we've got 6 patients total, 4 of them here at UCLA and 2 of them at Baylor.
Okay. And then, so from your sense on the way that it sounds like the vision that they have, and is the stimulator on the whole time?
So they get to decide when the stimulator is on or off. They don't have to wear the glasses or the VP, the video processing unit. You know, they can live their life normally without it. They will use it usually somewhere between 2 to 4 hours per day.
And the other thing too is that, so just so I'm clear, so how do you, so the surgical procedure is in 2 stages like DBS?
No, the surgical procedure is a single stage. So
Okay.
it's a U flap over the occipital lobe with a small parasagittal craniotomy to expose the interhemispheric fissure. The U shape is really so that we have skull exposure to implant the device, much like the NeuroPace device would be implanted on the skull. So it's a pretty quick procedure. And the first time we did it, it took about 4 hours because we're just trying to figure out what we're doing. The first time you ever do a surgery. The last surgery is probably about like 2.5 hours, skin to skin with craniotomy, implant, and doing everything else, which is still pretty quick for a new device. And
Nader, how do you use, so just educate me a little bit about the procedure. Do you use, so how do you know, do you use navigation? Like how do you know where exactly to put it?
Yeah, that's a good question. And it's something we have evolved. And honestly, myself and Dan Yosher, who's done the surgeries at Baylor that I was present for, have done used somewhat different techniques. I do use navigation because what we have, we have this electrode array that's hooked up to a soft, but it's a predetermined trajectory for the array. And so once you imagine you're looking interhemispheric, you pass an electrode into that space, you can't really see where it is. And so the only way to know where it is to measure how deep you're going to be on that ribbon and the direction of the ribbon. So if you know your depth and you know the trajectory or the angle, then you should know where your electrode array is. So I use navigation to determine where my craniotomy should be, and then also to determine basically like a DBS, the trajectory and depth, and get that electrode in the right spot. And the goal is really to be as far back on the occipital pole as possible to cover the above and below the calcarine sulcus.
And then do you wake them up and then like a DBS where you go, okay, or do you just do it and then wake them up from surgery?
We just do the implant. We thought about waking them up, but in truth, it's one of those things where you can't make that many adjustments. So you sort of get what you get, so you've got to get it right the first time. And so we decided not to wake them, that it would be discomfort that wouldn't really change how we're going to do the surgery.
That's really incredible work, Nader, that you're doing. and obviously you guys are publishing this, but I think it's amazing that, you know, just even with us, even though you have 6 patients, it's really fascinating to me that, you know, patients, just by stimulating their cortex, you can get some vision. It's remarkable that those connections are still there, you know, and working.
Yeah, that was one of our big questions, is because a lot of studies have been done in patients who are sighted, and we didn't know how it would translate to people who are blind or how consistent it would be. But it's worked out fairly well. And if, you know, people, the number one critique we get, especially from other people who work in this field, is, oh, but your resolution isn't that good. And I acknowledge it's not real vision. It's not the vision that we know. But again, if someone can, you know, the brain is a extremely powerful organ. It can learn. It can learn to reinterpret these signals and learn how to use them in a useful way. So we have to sort of break that mold of thinking we have to achieve perfection and perfect vision, but instead provide them with a tool that they can really use in ways that we can't imagine because we're not experiencing it.
Mm-hmm. I mean, the other thing for me too is, you know, just, you know, I've always thought of, you think of vision as being kind of, I think it's probably, like you pointed out in some of your slides, it's such a complex function. I'm sure in each of those patients, they've, you know, what you said about not restoring vision, but you're, you know, enabling them to kind of have some, you know, some sight. I wonder if how, you know, I wish there was a way you could kind of figure out what exactly that they're seeing, you know, because I'm sure it's probably different in every patient.
Yeah, they do. They have different perceptions and we don't know why.
Mm-hmm.
We're trying to understand that. We're trying to understand whether it's because of the location of the stimulation, trying to understand whether it's, there's just so many variables that it's hard to nail it down, but is it the cause of vision loss? Is it the age of vision loss? Is it the duration of vision loss? Is it the way we're stimulating? Is it the stimulation patterns? Is it the impedances? There's so many factors. Like we had one patient that for some reason his perception was that everywhere we stimulated was like, he saw a square, almost like a pixel with every stimulation, which is different than all of our other patients. And so it's tough. It has to be personalized therapy.
And then did you have any, I didn't realize, so when you did the study, were there any patients who were like blind since birth?
No, they're all previously sighted patients. And the
Okay.
reason for that is, you know, as we know from our neuroscience classes with the cat experiments, humans, we presume that's true for humans, but at least in cats, visual exposure is critical for development of the visual cortex. And so we wanted to sort of start with the maximum possible response, with the idea being, you know, let's make sure that their visual cortex is formed before we start implanting people who are blind since birth. Because we don't know, in people who are blind since birth, we don't know how their brain will respond. Hopefully, you know, if this all works out, that can be a future subject of study, which I'm sure there will be interest in that, but we're not there yet.
It's really exciting stuff, Nader, that you guys are doing. And as far as I know, do you know if there are any other trials around the world that are working on this?
There's a couple groups. There's a group in Australia that is working towards first in human trial. There's a group in Chicago that has also been working on a different type of approach. And then there are at least a couple European groups that are working on this as well. But in terms of actually having patients implanted, we're the only group that's actually implanted with a chronic device that has stayed in patients for more than 2 years.
Wow. That's really terrific, Nader. We're, to me, it's kind of fascinating. I know I saw some of our functional guys on I want to see if anybody has any questions. But you know, to me, it's what's incredible is all the stuff you guys are doing in neuromodulation, you know, with chronic pain, even, you know, epilepsy, and depression. I mean, I'm sure, what are the numbers like for depression? It must be like 100 million people. I mean,
well, there's a lot of people with depression.
yeah.
People who qualify for.
I mean, sorry, clinically, you know.
Yeah, clinical depression is
yeah.
I don't know if it's 100 million, but it's a lot.
It's got to be staggering.
Yeah, depression is an interesting field because there are a lot of potential solutions out there, especially with non-invasive stimulation. But I think for those who follow the field of brain stimulation for depression, there's a lot of promise in terms of the approaches that are being taken now. You know, we just started our trial, but have promising early results even with our trial already. So but it takes, you know, we were in a field where there's so many reports of, you know, I did this, look how much it helped, uncontrolled open label trials. And we've learned nothing from them, except that it might work if you do an open label trial. So I think the type of approach that we're taking here, and you see others doing, is really what's needed, a methodical approach, testing the stimulation, blinded assessments to develop therapies that'll get approved and are adoptable.
Well, and also too, I mean, I commend you because, you know, it's very hard. I mean, just think about just getting this through IRB, you know, and then also, you know, I mean, even though 6 doesn't seem like a lot, that's a lot of patients to get with a new device that's, you know, it's not even, it's like it's a miracle that you guys were even able to get that many patients.
Yeah, we had a huge response when we tried to recruit patients. Surprisingly, because I don't know if I'd want to be the first person to undergo a surgery like this.
And how did you get it? Was this through, did the FDA, was this like a humanitarian exemption?
So so there's two important things about the FDA. One is it was designated a breakthrough device, and breakthrough device gets special category or special consideration in terms of expedited review, and it also allows for more dynamic sort of approvals and processing and amendments to the device. But then it went through it's an investigational device exemption. So an IDE is how you do the first round of first-in-human trials, and it's an interesting process. We attended an in-person meeting of the FDA. The FDA, you know, they ask a lot of questions. The panel has people who cover everything from all different corners of consideration that you might not even think about, like electrochemical reactions, the safety, and things that are just not at the forefront of your mind. But they do a good job of getting multiple perspectives to make sure things are safe.
So did you have to go back to Maryland, or how did you guys do it?
Yeah, no, there have been a couple trips to Maryland. I went once for the full panel review.
Wow. No, I mean, just, you know, we do a lot, having been involved in lots of spine, you know, clinical trials. It's amazing. You know, people don't realize how difficult it is to, and, you know, in the United States, I think, you know, fortunately people complain a lot about the FDA and how long it takes, but, you know, I think they have a pretty remarkable group, and their system, they're so meticulous and methodical, and, you know, obviously safety is kind of the top priority for them. But it's amazing for me how quickly, you know, I know you've been working on this for like 10 years, but, you know, getting 6 patients for a trial like this is absolutely remarkable. So
yeah, I would say, you know, this has been like 6 or 7 years, but we went from literally concepts, from, you know, just talking over lunch to prototype, to study animal studies. Animal studies were done within like 2.5 years, 2.5, 3 years.
Mm-hmm.
And we were in humans in less than 5 years from concept, which is a remarkable speed, unless you're Elon Musk, in which case things can happen differently.
Oh, it's incredible.
Yeah, it happened very quickly.
Yeah. And then the other thing is, did they pay for, how did you do the reimbursement? Because it's obviously startup. They paid for the devices?
So it's been a combination of funding sources. To sort of get the trial up and going, that was all company sponsored. And then we actually applied to the BRAIN initiative for novel brain stimulation devices and have funding from the NIH now to support the study. So
wow,
it's a NIH-backed study.
That's great, Nader. I'm going to see if anybody else has any questions. I feel like I'm hogging you to myself. You know, for me, it's amazing. You know, I remember sitting in the conference room, you know, and I know you remember this as well, but, you know, those darn brain slices, you know, that we had to do with Dr. Jane on Saturday morning. It's incredible. You know, even when I look back, I'm always like, you know, oh, it's slide number so-and-so, and then this is the visual pathway. You know, there's the circuit of Pepez. It's incredible. I mean, you know, you can never go away from the anatomy. And it's as I get older, the more you think you know, the less actually you realize there's more that you don't know.
Yeah, I think that's totally right. I think there's so many, it's good to be optimistic when you start, but I've learned so much of what I don't know throughout this process.
Yeah.
And I think the important thing is to acknowledge what you don't know. And like I said, like you make certain assumptions. You know, we went to the anatomy lab. So we basically got cadaveric heads and we said, we got to figure this out. I'm a surgeon. I do things all the time. I just didn't know how this was going to feasibly work until I actually put it in a human head, to ensure that we were doing it the right way. So there's a lot of, I think it's just that, it's knowing when to say I don't know, and knowing when to say I need to ask more questions and to actually make something work.
Well, and I think also too, I think you have, I mean, I remember when we were medical students, you know, I think you've done it. I think you have to be able to get the neuroscience people, the bioengineering, and you got to get everyone to kind of, you know, think about what it is that you're doing. And I think this is a really cool example of a project where, you know, you're working with engineers, you're working with scientists, but then they all have to realize that this is real life, you know. You're not dealing with rats and mice. And, you know, for me, you know, this is what's incredible with some of the work that you've done, is be able to make that switch and apply it to, you know, everyday clinical problems. And I think you have a unique skill set, and very few people can do that. So I commend you. You know, this is really amazing work that you're doing, Nader. So keep it up.
God. Thank you so much for having me. And
yeah,
enjoy it. I know it's a little bit more scientific and less clinical, but in the end, this is all towards a clinical end. But it's just, there's so much that we need to learn. It's been a great experience, and I think it just shows that we can actually find solutions when we work hard with teams.
Yeah, no, definitely. And also, I think too, and I think you've seen this, Nader, but you know, there's so many cool things going on. Like in Seattle, we have the Allen Neuroscience Institute.
Yep.
And you know, they have the mouse, you know, brain. Then they have the human brain. You know, and I know they're super interested. I know Paul Allen was so interested in, you know, both visual and all these pathways that no one really understands. So I think the work that you're doing is terrific.
Thank you.
look forward to seeing you in person.
Yeah.
Thanks
Looking
again,
forward to
Nader.
seeing everyone in person. Thank you so much for the invitation.
And then just to everyone in the audience, you can get your, you can text to get your CME credit. And then I'm gonna put a plug in. We have a artificial discourse on Saturday. So look forward to seeing everyone there. And Nader, all the best to you and your family, and keep up the great work.
Thank you. Great to see you. Say hi to everyone.
Okay, you too. Take care.
Yeah, take care.
Okay, bye.
Thanks for watching. Hit the subscribe button for more medical content.